Systematics of Living Organisms. Can you recall what is five kingdom system of classification? There is great diversity of organisms around us. Since time immemorial, we humans have been exploiting this wealth for our own benefit. During this process, man tried to differentiate between and identify the organisms. Eventually, this evolved into a branch of biology known as systematics or classification. The method of classification dates back to ancient time when Indians, Greek, and Roman philosophers have contributed their might to systematize science. 2.1 Systematics. Systematics is the study of kinds and diversity of organisms and their comparative and evolutionary relationship. G. Simpson, 1961. Taxonomy. Taxonomy means classification following certain rules or principles. Word taxonomy comes from two Greek words, taxis meaning arrangement and nomos meaning law or rule. The term taxonomy was first introduced by A.P.D. Candolle, Swiss botanist, 1778 to 1841. 2.2 Classification. It is the arrangement of organisms or groups of organisms in distinct categories in accordance with a particular and well established plan. This classification is based on similarities and dissimilarities among the organisms. Artificial. It is the classification that is based on few easily observable and non evolutionary features such as habit, color, form, etc., often irrespective of their affinity with other organisms. Example, linear system of classification. Natural. Natural, it is the classification which is based on objectively significant rather than being selected for convenience like artificial system of classification. Example, Bentham and Hooker system of classification. Phylogenetic. It is the classification based on common evolutionary descent. Example, Engler and Prandtler's classification. In the system of classification, the term like taxa and categories are often used. Each category is referred to as a unit of classification. In fact, it represents a rank and is commonly termed as taxon. 2.3 Three domains of life. It is believed that the life originated on earth in its very simple form. Constant struggle of the early living being gave rise to more and more perfect forms of life. This struggle and progress is evolution which led to formation of diverse life forms. Carl Woese in 1990 proposed three domains of life to classify life forms. They are archaea, bacteria and eukarya. Domain is an unit larger than kingdom in the system of classification. Bacteria and archaea both have prokaryotic cells, whereas eukarya have eukaryotic cells. All the three domains have very unique ribosomal RNA, that is rRNA. Archaea are known for their survival in very extreme conditions like high temperature, salinity, acidic condition, etc. Bacteria, though are prokaryotes, differ from archaea in structure of cell wall. 2.4 Chemotaxonomy. It is the method of biological classification based on similarities and differences in structure of certain compounds present among the organisms being classified. In short, it is the classification based on chemical constituents of organism. Example, archaea cell walls is without peptidoglycan and that of prokarya is with peptidoglycan. Among eukarya, fungi have chitinous या फिर you can say chitinous cell walls while plants have cellulosic cell walls. 2.5 Numerical taxonomy. The system is based on quantification of characters and develops an algorithm for classification. The basic aim of this taxonomy was to create a taxonomy using numeric algorithm like cluster analysis rather than using subjective evolution of their properties. This system was first proposed by Sokel and Sneath in 1963. Cladogram. It is a typical branching pattern as shown on previous page, a diagram of three domains of life in a cladogram. It represents a hypothetical relationship denoting a comparison of organism and their common ancestor. Phylogeny. It is evolutionary relationship of organism. It is an important tool in classification as it takes into account not merely the morphological status but also the relationship of one organism of uh, with other organism of life. The system helps to understand the evolution and also focuses on the similarities of their metabolic functioning. Whose three domain concept as well as Whittaker's five kingdom system are very good examples of phylogenetic relationship. 2.8 DNA barcoding. DNA barcoding is a new method for the identification of any species based on its DNA sequence from a tiny tissue sample of the organism under study. It helps to study newly identified species as well as understanding ecological and evolutionary relationships between living beings. The process of DNA barcoding includes two basic steps. Number one, collecting DNA barcode data of known species, and number two, matching the barcode sequence of the unknown sample against the barcode library for identification. DNA barcoding has many applications. A few to mention are protection of endangered species, preservation of natural resources, pest control in agriculture, identifying disease vectors, authentication of natural health products, and identification of medicinal plants. Can you tell which characters of organism are visible characters? Name the recent approaches in taxonomy, what is DNA barcoding, what is evolution, and enlist uses of taxonomy. 2.9 Taxonomic categories. Classification is not a single step process but involves hierarchy of steps in which each step represents a rank or category. Since the category is a part of overall taxonomic arrangement, it is called taxonomic category, and all categories together constitute the taxonomic hierarchy. Kingdom, division, class, order, family, genus, species are the categories in hierarchical sequence. These are compulsory categories. Besides, there are some facultative categories like suborder, subfamily, etc. to be used as per need. 2.10 Taxonomic hierarchy. Taxon. A taxon is the taxonomic group of any rank in the system of classification. Example in plant kingdom, each one of the following such as angiosperm, dicotyledonine, polypetalae, malvaceae represents a taxonomic group, that is a taxon. Category, taxon, and the taxon. Kingdom, Plantae, Animalia. Division or phylum, Angiospermae chordata. Class, Dicotyledonine and Reptilia. Subclass, Polypeptalae, Diapsida. Series, Thalamiflorae. Order, Malvales, Squamata. Family, Malvaceae, Elapidae. Genus, Hibiscus, Naja. Species, Rosa sinensis, Naja. 2.11 Units of classification. Species. Species is the principal natural taxonomic unit, ranking below a genus and denoted by Latin binomial, considered as the basic unit of classification. It is a group of organisms that can interbreed under natural condition to produce fertile offspring. It was thought to be an indivisible, stable, and static unit. However, in the modern taxonomy, subdivision of species such as subspecies, varieties, and population are seen and gives more importance and is given more importance. Genus. Genus is a taxonomic rank or category larger than species used in biological classification of living and fossil organisms. Genus is a group of species bearing close resemblance to one another in their morphological character but they do not interbreed. Example, tiger, leopard, lion, all three belong to the same genus Panthera. They have common character yet are different from each other because their genus is same but species is different. Another example is genus Solanum. Brinjal and potato both belong to this genus. Family, it is one of the major hierarchical taxonomic rank. A family represents a group of closely related genera. Example, genera like hibiscus, gospeum, sida, bombax are included in same family Malvaceae. Cat also belongs to family of leopard, tigers and lions family Felidae, but dog belong to different family Canidae. Cohort or order. It is taxonomic rank used in the classification of organisms and recognized by nomenclature codes. An order is a group of closely related families showing definite affinities. Order thus is a step above family in taxonomic hierarchy. Member belonging to same order but different families may show very few dissimilarity. Example, family Papaveracea, Brassicaceae, Caparidaceae, etc. While parietal placentation are grouped in order parietales. Families of dogs and cats, though are different, they belong to same order Carnivora. Class. The class is the distinct taxonomic rank of biological classification having its own distinctive name. A group of higher taxonomic rank than order. Class is the assemblage of closely allied order. Order Carnivora and order Primates belong to class Mammalia. Thus, monkey, gorillas, gibbons, and dogs, cats, tigers belong to same class. Division or phylum. The division is a category composed of related classes. Example, division. Angiospermae includes two classes, dicotyledonii and monocotyledonii. In animal classification, division is a subunit of category or phylum. Subkingdom. Different divisions having same similarity form subkingdom. The divisions Angiospermae and Gymnospermae will the subkingdom Phanerogams or Spermatophyta. Kingdom. It is the highest taxonomic category composed of different subkingdoms. Example, subkingdom Phanerogams and Cryptogams form the plant kingdom or Plantae, which includes all the plants, while all the animals are included in kingdom Animalia. The taxonomic categories we have considered so far are broad categories. Scientists have added subcategories to these in order to place organisms in more scientific manner. You will observe that as we go higher in taxonomical ladder, number of common characters go on decreasing. If we are comparing two organisms that are related to each other, only at division or phylum level the classification may become difficult. 2.12 Nomenclature. Any object that becomes known to human intelligence must possess a name. It may not be possible or convenient to describe it in order to communicate ideas about it. The art of naming the object is in fact a science called nomenclature. All living organisms are known by a particular name. Number 1, vernacular or local name or common name. Widely distributed organisms have a large number of common names. Pansy, Viola tricolor L, grown in most European and American gardens, has about 50 common English names. In a multilingual country like India, almost all useful plants have local names which differ from language to language and even from dialects to dialects. As in Ayurveda, mango, Mangifera indica L, is known by over 50 different names, all in the Sanskrit language. Hence, the common names obviously have limited usage, and for universal application, a unique name for a particular individual is very much essential. Scientific names. To overcome the difficulties raised by common names, scientists have given scientific names to all the known organisms. These are systematic, thus provide means for international communication. Initially, the polynomial system was used by Carl Linnaeus, used binomial system of nomenclature. He introduced this system in his book Species Plantarum, published in 1753. International Code of Botanical Nomenclature (ICBN) has been set up to confirm the scientific names. Before 2011, the code which was set up to confirm the scientific names was ICBN, means International Code of Botanical Nomenclature. Recently, 19th International Botanical Congress (IBC) was held in Shenzhen, China in July 2017. This code is also called Shenzhen Code. So the old code ICBN has been changed to ICNAFP, means International Code of Nomenclature for Algae, Fungi and Plants. This code was published on 26th June 2018. According to this system, the scientific name of sunflower is Helianthus anus. In the above, Helianthus indicates name of the genus, generic name, and second word anus denotes name of the species. The binomial nomenclature system follows certain rules. Name of organism is composed of two Latin or Greek words. Generic name is a simple noun. It should come first and begin with capital letter. Specific name is the descriptive adjective which should come later and begin with small letter. Scientific name must be underlined separately if handwritten and must be printed in italics. The generic and specific name should not have less than 3 letters and more than 13 letters. Usually, the name of the author who names a plant or animal is also written in full or abbreviated form after scientific name. Example, Mangifera indica L, where L stands for Linnaeus. Carl Linnaeus classified living organism in two kingdoms based on mode of nutrition, whether they are migratory, sedentary, etc. But this broad classification as kingdom Plantae and kingdom Animalia was found inadequate. It could not classify the organism that show character of both the kingdom, for example, bacteria, fungi, euglena, etc. Hence, to avoid confusion, scientist R.H. Whittaker, 1969, proposed five-kingdom system of classification. This system shows the phylogenetic relationship between the organism. The five kingdoms are Kingdom Monera, Kingdom Protista, Kingdom Plantae, Kingdom Fungi, and Kingdom Animalia. 2.13 Salient features of five kingdoms. Number 1. Kingdom Monera. It contains unicellular organisms with prokaryotic cellular organization. Monera includes unicellular prokaryotic organisms. These are omnipresent. They are found in all types of environment which are not generally inhabited by other living beings. Few are photoautotrophs or chemoautotrophs, but majority are heterotrophic in nature. These organisms do not have well-defined nucleus. DNA exists as a simple double-stranded circular single chromosome called as nucleoid. Smaller circular molecules of DNA as extra chromosomal genetic element called plasmids are often present. Cell wall is made up of peptidoglycan, also called murine, which is polymer of sugars and amino acids. Membrane-bound organelles, example mitochondria, chloroplast, endoplasmic reticulum, are absent. Ribosomes are smaller in size, 70s, than in eukaryotic cells. The mode of reproduction in Monera is asexual or with the help of binary fission or budding. Very rarely sexual reproduction is by conjugation method. Morphologically, bacteria are categorized into four groups: the spherical coccus, the rod-shaped bacillus, the comma or kidney-shaped vibrio, and the spiral-shaped spirillum. On the basis of evolution, bacteria can be classified as archaebacteria and eubacteria. Archaebacteria. These are differentiated from other bacteria on the basis of their different cellular features. These bacteria are mostly found in the extreme environments, hence termed extremophiles. They are found in a variety of places from volcanic craters to salty lakes and hot springs. Their ability to withstand such hostile environments speaks of their capacity to survive in very severe conditions. Bacteria that can withstand high salinities are called halophiles, while those that withstand extreme temperature are known as thermophiles. A very common example is of methanogenic bacteria found in gut of ruminants, cows and buffaloes. These bacteria help in production of methane in biogas plants. Eubacteria. Uh, these are commonly referred as true bacteria. They have cell wall of peptidoglycan. They are found as autotrophs and heterotrophs. The autotrophs can be photosynthetic like chlorobium, green sulfur bacteria, and chromatium, or chemosynthetic like sulfur bacteria. These are mostly multicellular filamentous forms living in fresh water. The body is covered by mucilaginous sheath. The genetic material is typical prokaryotic. Chlorophyll A and chlorophyll B, carotenes and xanthanophylls are the photosynthetic pigments. Filaments show heterocysts which help in nitrogen fixation. Heterotrophs are the most abundant. Most of them are decomposers and known for breaking down large molecules into simple molecules or minerals. They can be anaerobes, helping in curdling of milk, lactobacilli, fixation of nitrogen, azotobacter, antibiotic production, streptomyces, composting and degrading oil. But the story doesn't end here. Some of them are pathogens, that is, causing disease like typhoid, cholera, tuberculosis, tetanus. Mycoplasma. These are smallest of the living forms. They do not have cell wall. Many forms are pathogenic. They are found resistant to common antibiotics due to absence of cell wall. Kingdom Protista. This group includes all the unicellular but eukaryotic organisms. These organisms show link with all eukaryotic kingdoms like plantae, fungi and animalia. Plant like Protista, they are all termed chrysophytes. They are commonly termed as phytoplanktons. They are microscopic and mostly photosynthetic and are major producers in oceans. Most of them are referred to as diatoms as they have body wall made up of two soap box like fitting silica covers. Diatomaceous earth is nothing but these shells left behind for so many years. Diatomaceous earth is granular, hence find use in polishing and filtration. Animal like Protista, they are also termed protozoans. They lack cell wall. They are heterotrophs. They are believed to be primitive animal forms. Amoeboid protozoans have pseudopodia as locomotory organs. Amoeba is free living form but entamoeba is endoparasite and cause amoebic dysentery. Flagellated protozoans have flagella as locomotory organ. Trypanosoma is a common flagellated pathogen which causes sleeping sickness. Paramecium is a ciliate protozoan having cilia for locomotion. In paramecium gullet, a cavity opens on the cell surface. Plasmodium is a sporozoan protozoa. It causes malaria. It forms spores in one of its life stage. Dinoflagellates. These are aquatic, mostly marine, and photosynthetic. The cell wall is made up of cellulosic stiff plates. They possess a pair of flagella. They have a wide range of photosynthetic pigments which can be yellow, green, brown, blue, and red. Goniolax is dinoflagellate that is responsible for famous red tide. It makes even sea appear red. Fungi like Protista. They are commonly from the group Myoxomycetes. These are saprophytic organisms found on decaying leaves. Their cell aggregate to form a large cell mass called plasmodium, not a malaria parasite. The spores produced by plasmodium are very tough and survive even very harsh conditions. Euglenoids. They lack cell wall but have a tough covering of proteinaceous pellicle. They possess two flagella, one short and other long. They behave as a heterotroph in absence of light, but possess pigments similar to that of higher plants for photosynthesis. Number 3, kingdom plantae. The kingdom is dominated by autotrophs. It also includes some semi-autotrophic members. The insectivorous plants like Venus flytrap, pitcher plant, bladderwort, as well as heterotrophic parasitic members like Cuscuta. Members of this kingdom are multicellular, having eukaryotic cell containing chlorophyll. Cells have cell wall mostly made up of cellulose. Plant exhibit alternation of generation, that is, life cycle has two distinct phases. Kingdom Plantae is divided into two major groups, cryptogam and phanerogam. We will study this kingdom in detail in next chapter. Number 4, kingdom fungi. These are eukaryotic heterotrophs showing extracellular digestion. They are found in warm and humid places. They have simple body which may be unicellular or made up of long thread-like structure called hyphae. Large fungi such as mushrooms have a compact mass of cells. Unicellular organisms have a protoplast with many nuclei, that is with many nuclei. Example, Rhizopus saccharomyces. Saccharomyces is yeast, unicellular fungus. Filamentous fungi consist of a body called mycelium in which hyphae are present. Hypha is present. The hypha may be with septa or without septa. They may be uni or multinucleate. The non-septate multinucleate hypha are called coenocytic hypha. The cell wall in fungi is composed of chitin, a polysaccharide or fungal cellulose. The fungi exhibit heterotrophic mode of nutrition. Mostly they are saprophytic. Some are parasitic or predators. They reproduce sexually as well as asexually. Asexual reproduction takes place by fragmentation, fission, and budding. Some fungi are symbiotic, either live with algae as lichens or as mycorrhiza in association with roots of higher plants. They are useful as well as harmful. Mushrooms are consumed as food. Yeast is used in bakery and breweries. Penicillium, a fungus, is well known for antibiotic production. Harmful fungi cause disease in plants and animals, example Puccinia. The fungi are further classified on the basis of their structure, mode of spore formation, and fruiting body as follows. A. Phycomycetes. These are commonly called algal fungi. Mycelium is made up of aseptate coenic hypha. They commonly grow in moist and damp habitats on decaying organic matter as well as in aquatic habitats or as parasites on plant. Example is Mucorrhizopus albugo. B. Ascomycetes. These are called as sac fungi. These fungi are mostly multicellular. Rarely unicellular variety include yeast. The hypha are branched and septate. Sac fungi can be decomposed or parasites or coprophilous, like grow on dung. Morels and truffles are varieties of sac fungi that are consumed as Delicacies. Neurospora is useful in genetic and biochemical assays. Example is Aspergillus penicillium claviceps Neurospora saccharomyces. C. Basidomycetes. These are commonly called club fungi. They have branched septate hypha. Example Agaricus, Ganoderma, Eustilago, Puccinia, etc. D is deuteromycetes. These are called imperfect fungi, which are known to reproduce only asexually. Example Alternaria, Coletotrichum. Coletotrichum. Number 5 is kingdom Animalia. Member of this kingdom are heterotrophs adapted to Holozoic nutrition. Most of them have capacity of locomotion. They are multicellular eukaryotes where cells lack chlorophyll as well as cell wall. Growth is determinate, follow definite pattern. In chapter 4 we will study about kingdom Animalia and its further classification. Viruses, viroids, are a group of acellular organisms that are not included in Whitaker's 5 kingdom classification. Acellular organisms. Viruses. Viruses were named so by Louis Pasteur, considering the meaning venom or poison. Venom or poison. These obligate parasites were given the name virus by M.J. Bijner, Bijner, Bijner after observation that they were able to migrate in agar gel. Thus, being an infectious soluble agent, he called the filtrate as Contagium vivum fluidum. It was scientist Stanley who demonstrated that viruses are inert outside the host cell and can be crystallized. They are made up of proteins. Viruses lack their own cell machinery. They have protein coat capsid around nucleic acid strand, thus considered to be acellular organisms. Viruses are inactive outside a host cell, but once they enter their specific host cell, they take charge of cellular machinery of host cell and duplicate themselves. Viruses thus can be called infectious nucleoprotein particles. Types of viruses. As per genetic material, viruses are grouped as DNA or RNA viruses. Protein's coat called capsid is made up of smaller unit, the capsomeres. Capsomeres are arranged in polyhedral or helical forms. Capsid protects genetic material. The genetic material in viruses is either single-stranded RNA or single or double-stranded RNA or double-stranded DNA. Viruses that infect bacterial cells are called bacteriophages, which normally have double-stranded DNA. Viruses cause disorders like leaf curling, yellowing, mosaic formation, etc. in plants. You have heard of foot and mouth disease in animals or swine flu, which are viral disease. Smallpox, mumps, herpes to common cold. Viruses are the causative agents of many diseases in human. The list includes AIDS too. Viroids. Potato spindle tuber disease was found to be caused by single-stranded RNA which lack protein coat. Theo Diner in 1971 reported that this is low molecular weight RNA and smaller in size than viruses. These infectious RNA strands are called viroids. Number 3 is lichens. Lichen is coexistence of algae and fungi for mutual benefit. Algal member, the Phycobiont as it is called, mostly belong to cyanobacteria, that is blue-green algae or green algae. Fungal member is called Mycobiont. They are excellent sample of symbiosis. The algal component of lichen provides food to fungal part while fungus provides shelter to alga and also absorbed water and mineral to alga. The association is intense and it is difficult to identify them as a separate living being. Though found in extreme environments like snow clad poles, lichens are sensitive to pollution. They are not found in polluted region, hence are considered as pollution indicators. Lichens also play important role in soil formation by using specific acid production. So this is the table exercise that you have to complete. You can refer to these questions. And thanks for watching the video. Please do like, share and subscribe my channel. Thank you.